Hello, everyone. My name is Cristiane Franca. I am a research assistant professor at the Oregon Health and Science University, specifically at the Night Cancer Precision Biofabrication Hub, and with a joint appointment in the Department of Oral Rehabilitation and Biosciences at the School of Dentistry. I thank you so much, the organization of Lab Roots, for accepting my name for this talk. I'm excited to share with you some of the accomplishments and discoveries that we've done in the area of microfluidics and organs on a chip for biomaterials testing and early cancer research. My goal here is to provide a fresh perspective on how we are envisioning cancer and how we are addressing several basic questions that can expedite the translational discovery of drugs for more targeted approach for cancer. Because as you know, more as we age, more and more people are being affected by and dying because of cancer. And this global cancer burden continues to rise. And it's the second cause of death in the US, just behind heart diseases. And we have this discrepancy here of we have more and more people having cancer, being diagnosed with cancer, and we still have this very slow and very expensive process of drug discovery for cancer. And the more we know about cancer, the more we know that we need precise drugs for each person, because cancer is more than 100 different diseases, and there is no one size fits all for cancer. So we are unique, just like cancers are. And we have this urgent need for cancer models that are functional, standardized, and equivalent or superior to preclinical animal models. Because now the standard way to study cancer is to develop everything in cell culture and then transfer the potential drugs to and test them in animals before testing in humans. There is this gap of the answers and the results that we have in culture, in animals, and in humans. And most of the drugs that are okay in animals, they don't work very well in humans. So we need humanized platforms that are fast, that can give big outputs, so we can test drugs in high throughput. When we think about cancer, we're thinking about a cascade of events that we need to break down all these events and different aspects to study them systematically, from the cancer initiation to invasion to the tissue, to intravasation into the vasculature, extravasation into the target organ, and metastasis. The focus of this talk today is going to be the steps 1 and 2, which is the early detection and the understand of the basic and fundamental questions of how to differentiate the lethal or indolent lesions very early. Just like if we have two babies, which one is going to be a terrorist and which one is going to be a good person, an entrepreneur, or some person that's going to be doing good in society, and intercept. What we want to do, our vision is that we can change, we can modify the environment in a targeted way, that these little cell that will has all the signaling and the genetic changes to become a cancer will not become a cancer. And this is what our body does. In fact, the body has this ability to heal and intercept cancer throughout our life, because in our entire life we're making cancer every day, and the body is intercepting cancer, and we need to understand how the body does that, so we can also learn with our own immune system, with our own body, to have interception of cancer, to decrease the burden of this disease in mankind. To that end, we need to start with baby steps. We microengineer the tumor microenvironment in early cancer. Here is a 3D modeling of the vasculature, the cancer, the collagen, the extracellular matrix, the immune cells, everything is happening. It's just like having this huge party with lots of people, hundreds, thousands of people, but each one has a specific role in this party, which is named cancer. So the aspect they are going to focus in our research, in our talk today, one of the projects is to study how cancer cells, they talk, their interplay with the extracellular matrix to become cancer. We all know, it's very known in the literature, that the extracellular matrix, the collagen, the other proteins, when they change, they can promote tumorigenesis. They can promote cancer to evolve from a benign lesion to an in situ carcinoma to invasive carcinoma. We can see that this figure is very fortunate to depict that the collagen that's very reticular and thin fibers here, it starts to condense, to increase its density, and it starts to align. We call it fibrillar collagen. In fact, every collagen 1 and 3, many collagens are fibrillar because they are formed of fibers. But here we're taking this term as we see in the radiographic exams, mammographies, for example, or in the histology exams. We call it, remember that this collagen has thicker fibers. And it's very difficult to mimic these in vivo, in animals, and to explore this part, this specific aspect of the collagen transformation during early tumorigenesis. There was this paper that was published in Nature Communications in 2015 that the authors found a way to gradually control collagen fibrillogenesis to get from these highly reticular soft fibers to this thick fibrillar bundled structure. The thicker the fibers, the stiffer they were. So this is exactly what happens in our body from the benign tumor, which is this related to this collagen here, to this specific conformation and architecture of the collagen, to this collagen here that stays in the tumoral front that we have here. That's the stiffer and more fibrillar bundled collagen. What does it make to the cells? Imagine that you have these very rigid ropes that the cells have to crawl over it. So the cells, they rearrange their anchorage system in stiffer environments so they can migrate faster, which is named durotaxis. And this changes several pathways related to cancer in cells. Usually the cells that are canonically related to these phenomena are fibroblast, cancer-associated fibroblasts and macrophages, especially M2 macrophages, polarized into a more pro-anti-inflammatory macrophages. So many of these cells, many of these changes, they are related to cancer-associated fibroblasts and macrophages. However, vasculature is a very important component of the tumor microenvironment. And even though we know that endothelial cells, they're mechanosensitive, how they interact with these tumoral vasculature and with this increased collagen fibrillogenesis is not very well understood. To fill this knowledge gap, we fabricated this microfluidic device. This device was developed by the group of Professor Christopher Chen from Boston University. And here's the citation that we used to develop this platform. However, what we did, we added collagen here and we changed the temperature of fibrillogenesis to get this gradual change of the collagen with increased stiffness and thickness as we decreased the temperature. You can see here we have a very highly reticular soft collagen, which we call healthy group. We have gradually an increase in thickness and stiffness of this collagen. We seeded the cells inside the channel, inside this collagen, this hollow collagen channels, and we're able to engineer single channels that are, biologically, they work like capillaries. However, what we saw is that endothelial cells alone, they mimic very poorly the vasculature. We don't see the changes that we see in tumoral vessels here with only the endothelial cells. Even the cell-cell adhesion are basically the same. So I say, well, what may be wrong? How can we improve this system, this microphysiological system, to make it actually microphysiological, to make it more predictive and emulate better the vasculature that we have in the body? There is one fundamental component that was not being considered, the perivascular cells, or for capillaries, the pericytes. Because in our bodies, in vivo, we all have vasculature that's surrounded and supported by capillaries, by pericytes. These pericytes in healthy vessels, they're attached, they're very, they have an intimate association with the endothelial cells, and they even share the same basement membrane. So that's why you can see here this green endothelial cell colocalized with pericytes. But when these cells are inflamed, when this vasculature is inflamed, we can see that these pericytes, they become activated and they detach from the vasculature, leading to a leakier vasculature. This is very well described in inflammation, but do these pericytes sense the mechanical changes in the extracellular matrix and then transfer them to the endothelial cells? Because what we can see from our previous slide is that the endothelial cells, they sense, but they don't change very much from the soft and the stiff, between soft and stiff. And here, but we know that the vessels, the tumoral vessels, they're leaky. They leak and they have less pericyte coverage. So what is in this system that we're not mimicking? And we found it. We seeded these channels with aquaculture of endothelial cells and MSCs, mesenchymal stem cells, and they formed beautiful vessels when the collagen was soft and highly reticular. However, they were increasingly forming dysmorphic vasculature with more migrating cells, and these vessels were larger in diameter. They were heterogeneous in their morphology, just like tumor-like vessels, and they were leaky. You can see in this movie here that when we inject dextran in these vessels, these ones here, they don't leak. Normal vessels don't leak. When you have a slightly stiffer collagen, the perivascular cells, the pericytes, they are capable to maintain the barrier function of the vasculature. But after a certain stiffness, which is not very high, we're talking about 5 kilopascals only. We're not talking about 40 or 40 kilopascals as some tumors have. As the stiffness and microarchitecture changes, the vessels become leakier and leakier, mimicking a tumor-like vessel. And here you can see the difference between this normal vasculature with this homogeneous cylindrical shape and these migrating cells, but most likely you have these red cells as the CD31, which marks an endothelial cell-cell junction. And in magenta, you're going to see the perivascular cells attaching to the vessel, some is slightly migrating, which is compatible with a normal vessel. And here, what you're going to see is a tumor-like vessel that we engineered without any cancer cell. We engineered it only by changing the collagen stiffness and architecture and adding the perivascular cells in this system. And we can see that we have heterogeneous cell-cell contact. We barely have pericytes, and we have lots of migrating cells that are going outwards, outside the vessels. Well, we saw that the vessels, we can engineer the spectrum of vessel morphology differently, and we can engineer the spectrum of changes in barrier function of the vessels just by changing the collagen. Our next step was to understand what mechanisms and pathways were being activated by the presence of perivascular cells in a stiffer microenvironment. And we could see that we have different, we did a nanostring. For this, we analyzed 770 genes, and we could see that genes related to CXL8, genes related to pro-inflammatory, paracrine signaling, proteoglycans, as you can see the decorin, remodeling of the extracellular matrix, TGF-beta, are all collagen 3 are all upregulated in a stiffer collagen, but that you have the MSCs there, the pericytes there, but are downregulated the genes related to angiogenesis in healthy vessels. So giving us some clues of what is happening to this tissue. We can see that pathways in cancer that are completely upregulated just by having a stiffer and more fibrillar collagen in contact with the vasculature and the perivascular cells. This doesn't happen when you have only endothelial cells. And we have pathways related to angiogenesis downregulated in normal vessels, which give us some hints of what are the potential targets for some anti-angiogenic therapy. So our next step was to block some genes in pericytes. We had the hypothesis that the mechanotransduction mechanisms would be increased and the cells would secrete paracrine signaling and paracrine signals to promote the vascular dysfunction. So we blocked integrin beta 1, we blocked Notch3. Notch3 integrins are responsible for the cell attachment to the extracellular matrix, to the collagen. Notch3 is responsible for the heterotypic cell-cell adhesion, where the pericytes, they are attached to the endothelial cells. And TGF-beta, as we all know, it's involved in the paracrine signaling of many processes related to both things. A double-edged sword, as I say here. It's related to either attracting pericytes to the vasculature and attachment of pericytes, but also it's related to epithelial-mesenchymal transition, cell migration, and many cancer pathways. What we can see is that when you block integrins, the normal and healthy vessels, they look like the fibrotic vessels, and they're similar in number of pericytes and number of migrating cells to the fibrotic vessel. So these vessels become diseased as if they were fibrotic when they stop sensing the regular softness of the collagen. When we block the Notch3, it means we're blocking the cell contact from the pericyte endothelial cell contact. What we're seeing here is that the vessel that were supposed to be dystrophic, in fact, normalizes. Same happened with TGF-beta. So blocking Notch3 and TGF-beta 1 can normalize the vasculature. From this work, it generates some evidence that pericytes are a novel potential target for anti-angiogenic therapies. We're in the process of running more experiments, and we want to test this hypothesis in vivo as well to see, but we're able to approach anti-angiogenic therapies under a new perspective, just doing the basic science and organs on a chip. Now, we're switching gears to another type of invasion of bone. It's not vascular invasion, but also bone invasion. Cancer, especially head and neck cancer, it has very anatomic proximity with bone. We have this epithelial here, we have this connective tissue underneath the salivary glands, and we have bone very close. It's 1 or 2 millimeters apart from each other. So many cancers, especially the head and neck, they invade bone very early in their trajectory, in their life cycle. So the patient and whenever the tumor has invaded bone, the prognosis decreases drastically because it's very hard to control a tumor that has already invaded bone. Bone is a very complex tissue. We have the extracellular matrix, which is mineralized to the nanoscale. We have blood vessels, capillaries, we have osteocytes, osteoblasts, and osteoclasts. All these, and in a lot of exchange of paracrine signaling in a remodeling, it's a very dynamic tissue. There are two current paradigms in cancer, head and neck cancer invasion to bone. First, these interactions are mostly osteoclast dependent. It means cancer cells arrive here, they activate these osteoclasts via paracrine signaling. These osteoclasts, they degrade bone. When they degrade bone, bone releases the growth factors that are entrapped in their matrix. And these growth factors, they give they're captured by the cancer cells, and the cancer cells become fueled by these growth factors and enter into very aggressive state. So this is classically known as the vicious cycle of bone metastasis, and I would say bone invasion here. We're not talking about metastasis, we're talking about steps earlier. And head and neck cancer invade bone due to anatomical proximity. It means the tumor is there, it happens in the mucosa or pharyngeal mucosa, the tumor grows, and it happens to have bone in the middle of the way. So it invades bone. Yeah, it can be, but if it were just like this, these drugs here, the drugs that address specifically some stage of the life cycle of osteoclasts would work for most of the patients, which is not true. Bisphosphonates and other monoclonal antibodies that are targeted for osteoclasts, they control bone metastasis and bone invasion, but they're far from being ideal to treat and cure or intercept a cancer invasion into bone. We want to break this paradigm and understand what if there's so many cells and so many things happening in bone, it's such a dynamic tissue, why are we going to target 1 or 2 cell types? What are the other cell types doing in cancer to promote this cancer invasion? So the group built on a work published on Nature Communications in which Dr. Bertassoni, Luis Bertassoni, he developed this rapid method to mineralize and vascularize bone to the nanoscale. It means the cells at the scale that matters, the scale of cell junction, the cell adhesion system to the bone, they sense the stiffness of that mineralized collagen fiber in a way that it triggers all the mechanisms for them to become osteoblasts, MSCs become osteoblasts and osteocytes without the need for growth factor, just by giving the proper environmental cues to these cells. So we built on this work and we mineralized the collagen, we encapsulated cells in the collagen, mineralized this collagen using this polymer-induced liquid polymerization, which is published in this paper here. And we did that in a chip that has 2 chambers, lateral 2 chambers in which we add cell median cells, And in the middle, in this blue area here, we have our collagen hydrogel that's mineralized for 3 days. And what we have is that with osteoinductive medium, we achieve some degree of mineralization. But with this method, to add calcium phosphate, and we can have more mineralization in 3 days. And we have this proved by the alizarin red staining and von Kossa staining. And this is the non-mineralized collagen as our negative control. The cells that are encapsulated, the mesenchymal stem cells that were encapsulated in this bone, they differentiate into bone-forming cells, osteoblasts and osteocytes, in a way much without the need for growth factors, in a way that's much better than using osteoinductive medium. And we call this biomimetic mineralization. And we can do that in a controllable manner on a chip. So we use this platform to understand what is the effect of the other bone cells, osteoblasts and osteocytes, in attracting the cancer cells into bone. Is this just a question of proximity, or are there other paracrine signaling in place that we are overseeing? So we seeded the collagen with no bone cells. Here is acellular collagen. And this collagen here with bone cells, which means with osteoblasts that later will differentiate into osteocytes. So every time that we say about bone cells, is osteoblasts with osteocytes. And what we can see right off the bat is that when we seeded the same number of cancer cells, and look in this lateral channel how many cancer cells we had here. So we have more cancer cell proliferation when we have bone, cellularized bone, but not mineralized. When we have it mineralized, we have more cancer cells than we had when we don't have any cells here. But these cancer cells, they have a different shape, which called our attention. When you look at them closer, we can see that they express vimentin, but these are epithelial cells. Epithelial cells express vimentin when they're undergoing into what we call epithelial mesenchymal transition, EMT, which is one marker of aggressiveness and metastatic potential of the cells, which means that the bone cells itself, without in a mechanism that's independent from the presence of osteoclasts, they, just by the paracrine signaling, they are inducing these head and neck cancer cells to undergo epithelial mesenchymal transition. This happens in this cell medium, this environment that is non-mineralized with no cells. These cells have a polygonal shape, but they still remain a little bit epithelial, as you can see here. But when they have the presence of bone cells, osteoblasts, both groups, mineralized and non-mineralized, they present epithelial mesenchymal transition. So there is this crosstalk between osteoblasts, osteocytes, and cancer cells that attracts these cancer cells to the bone. And these numbers we quantify these cells, and these numbers are very significant, which means that what is their talk? What are they saying to each other? We did this Luminex assay to track the most important cytokines and growth factors related to bone invasion and to bone modulation. And we can see the presence of osteoblasts and osteocytes downregulate the inflammatory cytokines in the presence of cancer cells. All groups were seeded with cancer cells, and the difference was there was no cells here in the matrix, but there were cells here in this matrix. One matrix was non-mineralized, the magenta one, the pink one is non-mineralized, the green one is mineralized. we can see that CXL5, MIP, MIP1, CCL4, TNF-alpha, interleukin-1 beta, MMP9, RANKL, RANKS, they're all downregulated, which is aligned to what we see in the literature, that bone cells, they have this immune modulatory potential. So they also regulate inflammation. But in the presence of cancer cells, these osteoblasts and osteocytes, they secrete much more interleukin-6. Osteoblasts are capable of secreting interleukin-6, and they secrete CCL2. Both cytokines, they are related to cancer invasion to bone, and they are related to the increase of some signaling related to STAT3, which is also related to epithelial mesenchymal transition. So this can be a potential mechanism for which we can understand how bone cells attract cancer cells. So 2 of our questions were answered by in the works to be answered by this project. The first is, are there mechanisms that are independent, osteoclast independent, that can attract cancer cells to bone? Yes, there are. Osteoblasts can attract cancer cells to bone by producing interleukin-6 and CCL2. And are these mechanisms targetable? Yes. And the novel thing is that osteoblasts and osteocytes, they can contribute to the epithelial mesenchymal transition of cancer cells. Just their paracrine signaling, just the proximity makes cancer cells become more malignant, independently of the remodeling of bone in the osteoclasts. So these osteoblasts and osteocytes, they promote an osteoclast-independent mechanism for bone invasion by inducing this EMT in head and neck cancers. And that can be a potential venue for more targeted therapies and for precision differentiation of lethal from indolent cancers. I want to thank the amazing team of the Bertassoni Lab, Luis Bertassoni. He was my mentor in my postdoc. He's my mentor in my transition to independence. And I want to thank him very much for the support, all the amazing team of his lab, and our collaborators, Christopher Chen, Michelle Kuhn, and Jack Ferracane from OHSU, and all the agencies that have been funding this work. So I want to thank you all for being with me for this talk. This is my email. Feel free to reach out, to contact, to exchange ideas so we can make more and more exciting science around. Thank you very much.